Good afternoon. I'm Ed Guo, Chair of the Department of Biomedical Engineering at Columbia University. Today, we are very excited to continue our research spotlight series, BME Faculty in Focus, where we will highlight the exciting research conducted by our Columbia BME faculty. So moving forward, these lectures will be available on our BME YouTube channel at youtube.com/Columbia BME. Throughout the presentation, if you have questions for our speaker, please feel free to type them into Q&A area at the bottom of your screen. We will try to answer as many as possible at the end of the questions. At this time, I'd like to introduce our BME faculty in focus speaker today, Professor Simon Sear. And I was just chatting with Professor Simon Sear, and then we met almost 20 years ago in Boston when I was doing my sabbatical and Sam was doing his postdoc. And Dr. Sam Sear developed technologies for point-of-care blood tests, wearable sensors, and implantable devices in both academic and the industry settings. He co-developed the point-of-care blood test for prostate cancer, which is being commercialized with FDA approval, and is a co-founder of Rover Diagnostics, which develops rapid, low-cost detection of DNA and RNA. He founded Harlem Bioscope, a biotech incubator facility in New York City. He has a Bachelor of Science in Chemistry from University of Alberta and a PhD in biophysics from Harvard University, and completed a postdoc fellowship in chemistry and chemical biology at Harvard University. That's where Sam and I met. Welcome, Simon. It's all yours.
Great. Well, thanks, Ed. So excited to be giving this presentation. And those of you watching this live, thanks for taking time on this beautiful Friday afternoon in New York. I'm going to put on my share screen here. I hope you can see this okay. And I'm sure Alexis will.
Yes.
Okay, great. That looks good. Okay, well, I'm already messing things up by changing the title, but actually our chair went over some of the focus areas in my lab already. And yeah, talking about wearable and implantable miniaturized devices, both for diagnostics and therapeutics. And I'm going to do this in 4 parts here. I'm going to start off with some of the diagnostics work, which we have been working on for a while, but really moving into maybe more futuristic areas where you want the testing to be a little bit more non-invasive. So the wearable devices will try to get us towards more everyday sort of monitoring. The therapeutic angle I also want to bring in, because these miniaturized devices can also help with that. And then finally, actually connecting the sensing with the therapeutics and really make it a closed loop system, which you can also close the loop using things like machine learning. I'm going to have a bit of a bonus 5th topic here, talking about the entrepreneurship space and touching on something called the 4th purpose, which is something I'm involved in at Columbia as well. So that will be towards the very end. Starting with the diagnostic and sensing work. This is a review that we wrote 6 years ago in analytical chemistry, reviewing some of the work back then on the trends of combining microfluidics, which allows you to do testing that you normally have to do in a big lab, but miniaturizing the fluid handling. But combining that technology with consumer electronics, hardware, software, user interface, wireless communication, and really combining those 2 areas of work, which traditionally haven't really been combined, can really bring us further towards this digital connected health age. Of course, this was before COVID, but we had outlined some thoughts on this with different use cases, even within the so-called point of care space. This is all point of care, by the way, but what we were saying is there's actually very distinct classes of point of care. And as a result, the way you design the devices actually have to be quite different because there's a spectrum of different point of care settings. Going over some of the work we did before, we worked on things that were sort of more lower cost, more in the field, that kind of use case. We worked on diseases testing, infectious disease testing, HIV, sexually transmitted infections in places like Africa, and published work along those lines, and then came up with a version that worked with a smartphone device. So you can do it very easily with all the advantages of really combining with the consumer electronics, with microfluidics. We tested this device again in sub-Saharan Africa because the device itself is actually reusable, is about $30 US, and then the per use cost is actually even lower, just for a few dollars per test. And then you can reuse this so-called dongle. That's tested in Africa. There's a device that we have been working on that I helped work on, but it was commercialized by this company called Opco Health, and they did it for the US market for prostate cancer monitoring, measuring quantitative levels of prostate-specific antigen and really other things that from a drop of blood from your finger, and this is something you usually need to do a blood draw, send it back to a lab. You need to have sensitive detections down to picograms per mil even, and it has to be quantitative, and that's challenging. But with microfluidics, you can actually try to do that in a doctor's office in something along the lines of a toaster oven kind of size, and they produce some great results. This is from them, not from our group, but compared with state-of-the-art centralized lab testing, you get really good correlation. Again, this is actually quantitative. Okay, so you can actually get that with the instrument that's called Claros here compared to Roche and Abbott big instrument tests. So yes, you can do this. Now, this is in a doctor's office. We tested a smaller version in Africa. This is all before COVID. Just a few months ago, we wrote a sort of a more updated review, and this is published in the journal Lab on a Chip. And I encourage everyone to try to look at that as well. We have reviewed in this paper multiple, really all the tests that have been EUA'd, and they're of different classes. Some of them are nucleic acid based, which you would be familiar with for the lab-based PCR test, but there's other types of nucleic acid tests, such as isothermal tests. Some of them are point of care. There's antigen tests, which you are familiar with for the rapid tests. There's also antibody tests that are also in that same form factor. So we review all of those in this review and talk about sort of the factors that are involved here to make it successful, as well as the trends that we observed during the pandemic, as well as the implications moving forward beyond the pandemic for future infectious disease testing, as well as just for digital health in general. And for COVID testing, we actually pointed out that the over-the-counter testing approved by FDA, so it's not just tests that are approved, but ones that are approved for over-the-counter use that you can actually purchase in, say, a pharmacy, have been rising all the way up until the pre-COVID age. And so this trend of increased decentralized testing by untrained users has been on the rise for quite a bit, and now it's being accelerated greatly during the pandemic. And right now we're in this mode called EUA. It's not the full-on FDA trials that you would need. It's a bit of a emergency scenario. And you can see that this has been done before in this emergency use authorization, but for COVID, the number of tests actually authorized here have dwarfed the number of EUA tests under other scenarios in the past. So pretty interesting. We also mapped out different. This is more of a business analysis. Most of the review is scientific, but we did look at the businesses that were involved in the COVID tests that have been authorized and made some observations, which I won't go into here, in terms of when they were founded, how much federal funding they needed to get it to EUA, with, again, some implications for what it takes to really stand up a new diagnostic test during an emergency. And of course, a lot of times you need to piggyback on technologies that have already been developed. Otherwise, you're not going to be able to develop it from scratch in an emergency. And yes, getting some help from federal funding would be important, but lots of lessons that are still being learned and digested for what this will all mean for moving beyond COVID into this era of digital health. And everyone has talked about point of care testing for a long time, but the COVID pandemic is really accelerating that trend, not only for COVID, but really for digital health. Now back to some of our own work, we are also working on some of these newer technologies. One here is a point of care PCR test. So you don't have to choose between getting the gold standard test that requires you to do a swab, send it back to a lab, or have a home test, but one that is, maybe a rapid test, but not as accurate. We think you can eventually do both. You get both of those things by having a home test, but a true blue PCR test. There are some home tests that are nucleic acid based. They're not PCR, but they're pretty good. But we think, but they're pretty expensive. They're not PCR. They work okay, pretty good, pretty well. But the big problem is they're still very expensive. And so we think we can offer a low-cost PCR test that could be even a home-based test at some point. And this is the startup company that I co-founded along with our founder, CEO, Mark Fasciano, and it's now Rover Diagnostics, actually taking up space in an incubator that's in the Columbia Medical campus. And they're developing the work as well. And we got support from the Radix program through the Radix Tech program, which is for the more mature technologies. And this is unpublished results here, so we hope will be published soon. But we've collected a lot of data where we think we can perform a very fast, well, I should just say fast, let's just say around, 20, I think it's around 25 minutes at this point compared to typical 45 minutes. We think we can actually get it down to much faster than that, but with the data that we've collected, let's say 25 from sample collection to result, and do a full-on RT PCR test that will actually give you CT values as well. And you can actually do real-time monitoring along with that. So it's really like what you would do in the lab. You're really not missing anything there. It's also multiplexed. And here we're detecting 2 targets for COVID, as well as a control for human to give you that positive control. And our limited detection here is somewhere in the range of thousands of copies per mil, which is kind of what you expect in a lab. So with clinical specimen testing, et cetera. So I won't go into all the details, but this is hopefully some results that will be public at some point. Now, through another program, we have been collaborating with Anne-Katrine Ullman at the Medical Center, as well as Kartik Chandran at the engineering school, wastewater expert, on doing wastewater monitoring. This is through another Radix program for sort of more early but innovative technologies called Radix RAD. And what we're hoping to contribute here with in this project is to simplify the sample processing. So right now with wastewater monitoring, you have to bring back the wastewater again back to a lab. And wastewater, it's not like clinical specimens. You have to collect, huge volumes of that, relative, like liters, relatively speaking, is a large volume. And so you have to centrifuge it down. There's manual steps, you're limited by centrifuges. And there's a bottleneck in the lab. So what we're hoping to do is to simplify all of that and really to actually automate it as well. So we can actually then have this sample processing instrument really just put in line in buildings. So you don't really have to do the sample processing back in a lab. This will be automatically be done for you in these building sites. So the person just comes and picks it up. Actually, what we want to do is actually have the detection to take place right there as well. So nobody even has to go to these sites. You don't have to pick up anything. You don't actually have to run anything. You can have these remote monitoring sensors placed into whatever buildings you want, whatever wastewater streams you want, and you will just get a real-time dashboard sitting somewhere telling you what the COVID situation is. So this is what we're trying to do with Anne-Catherine and Kartik, both in automating the sample processing for wastewater, and then piggybacking on some of those amplification technologies I showed you with microfluidics, also doing that part at the point of use as well. Speaking of point of use, that's something we have been focused on for quite a while, really going into design thinking of engineering. So back to our infectious disease sorts of point of care testing, we also came up with frameworks for actually even how to ask the right questions for users, going into their motivation, their information, and their behavior. This is based on behavior models from other fields that we synthesized for point of care diagnostics. And so we came up with a framework published here in Lab on a Chip and applied it to point of care diagnostics. We also applied this kind of thinking to rapid tests, so not microfluidic tests, but these rapid tests for example, sexually transmitted infections. And we worked with this with Ivan Balan, who is a behavioral expert. And you can see here, we actually surveyed a group here that is impacted by sexually transmitted infections. These are men who have sex with men, MSM population, typically not always speaking English either, and really have a population that we really need to reach if we want to make an impact for HIV and syphilis infections. But we need to think about what their behavior is if we are to successfully reach them, including the user interface of how we would ask the questions and ask them to run the tests. And so we ran this trial and really assess the usability of our app in encouraging them to test not only themselves, but their sexual partners, in encouraging them to really do everything safely. And we want to effect positive behavioral change with a digital tool like this. And so we want to understand this. We want to actually have some impact in the real world. Now, that was HIV and sexually transmitted infections. COVID came along and we decided to further develop that effort with COVID tests. And this is an effort we developed with Shifu Chang, who is our dean right now in the engineering school, but also an expert in image processing and machine learning. And what we decided to do was to automate the scanning algorithm. And subsequently, we ended up working with the Mayo Clinic, and we licensed this machine learning algorithm to a company called Safe Health. And they're a telemedicine platform. Now, what the idea here is to automate the scanning of these rapid tests. So like these rapid antigen tests, which a lot of you have probably run now for COVID. But, and you can use machine learning to read the lines of the tests, automatically interpret that for you. Why is that interesting? Because the app, as well as the automated interpretation, would make sure you're interpreting the test result properly. It might seem trivial that, oh, there's a band there, I can read it. What's the big deal? With a lot of the general population, it's actually not easy. You have to actually learn that there's a control line that has to be there. And is there a band? Is there not a band? So using this app, it would automatically interpret the result for you. Now, what Shifu's algorithm really enables one to do which is really powerful, is we can learn how to do that for any new test kit platform using only 20 new images of that specific rapid test kit format, which is different. There's different numbers of bands, they're all in different formats, and using traditional methods, you would need at least thousands of these validated images to train properly, because there are shadows, there's all this stuff going on. With this method, which is using something called few-shot learning, we train a base model using a large number of trained images, but then we can rapidly adapt to any new test kit format using only 20 new images. And this is going to be really important in the pandemic, where it's a lot of work and really not feasible to be collecting thousands of images for every new test kit that you have. And so we can train this for one base set of images, and then for any new tests, we can actually rapidly have this working as well. So for example, this might be the number of actual test images you would need to collect to test something, to train your algorithm de novo. It's a lot of images, but once you do it once, you only need a very small number of new images for every new test kit. And we can get very high accuracy with this method. And so there is a preprint that is out already if you want to take a look at it. We also did a field trial with a COVID site where people took the images in their own cars. So pretty realistic. Uploaded the images, and again, our algorithm could rapidly been trained to interpret both antibody and antigen tests accurately. So we'll be hopefully publishing that more extensive study as well. So really moving a lot of stuff with in vitro diagnostics, microfluidics, rapid testing for diagnostics. But like I said, we're also doing work on now to make things a little bit even less invasive, a little bit more futuristic. So I want to move into that work into the wearable devices. It would be great if you don't need to actually prick your finger, to draw blood, to do some of these tests. Doing a nasal swab is not a big deal, so that's great, but you don't get most information like that. You usually have to get it through blood. So what we're doing here is we want to develop a method where you can apply a patch onto your skin, and that would then more or less continuously monitor for the analytes in your interstitial fluid. It's not blood, but your analytes in your interstitial fluid typically track what's in your blood pretty closely, and there's a lag time. It's not always the same, but it's pretty close for most of the analytes. So we're doing this optically here. We're basically using microneedles. We're calling them microfilaments because it's not a hollow, hard microneedle. It's a soft hydrogel filament that's not hollow. The whole hydrogel that we embed within your skin has aptamers in it that will fluoresce in real time, depending on whether your analyte is present or absent. And then we would actually read this fluorescently. Most of the microneedle approaches out there use hollow microneedles. They actually suck the interstitial fluid out. There's some clogging, reproducibility issues there, and they do an electrochemical detection. We're doing this a little bit differently, where we don't suck anything out. We're just reading things through the transparent hydrogel in real time and monitoring the analytes. And so we came up with some just more fundamental polymer work here published initially. This is what these microfilaments look like. In their dried state, even though they're hydrogel, they're actually up in the megapascal range, and they're sharp, they can penetrate your skin. Looks scary, doesn't actually hurt very much. And your skin heals pretty fast right after they are removed because the microfilaments are so small. We did a pain test assessment for human subjects, and they report that they're it's kind of like rubbing Velcro on your skin. It's not like sticking a needle or an array of needles into your skin. And again, your skin actually heals even in live human subjects pretty fast. And so by using hydrogels, we think we can do that. And right now we're working with Michelle Rubin on applying this to calcium sensing for conditions there with hyper and hypocalcemia. And so here we go. And also with Milan Stojanovic as well. We have an R1 on this collectively on applying the technology for this setting. Another work that I'm also hoping will be published soon that I'm very excited about along the lines of a wearable device is more of a hardware wearable. It's kind of like an Apple Watch or a Fitbit, or in this case, it's going to be probably an armband, but we'll see. Something that can measure blood pressure really well. And really, there's no accurate way to measure blood pressure without a cuff. There's devices out there, they're not, they don't give you accurate absolute numbers. One of the reasons is that you need to correct for what is called the hydrostatic pressure. A lot of these blood pressure devices require you to take your blood pressure at the height of your heart, because otherwise the blood pressure varies hugely depending on the relative level compared to your heart. That is something that is basically uncorrected in almost all of these devices, and therefore they can't give you the right number. They can only give you a relative change. So Alex Colburn, very talented former PhD student in our lab, really his project, he came up with a way, which I'm not going to talk about in detail here, on how to correct for that. Really clever. We parameterize arm movement using Eulerian angles, use machine learning, which would not have worked without the parameterization because it is too unconstrained a problem. But with some hardware and software and also parameterization, physics-based, we were able to automatically detect what your arm orientation would be, which lets us correct your blood pressure. So here's some human subject data where they can do whatever they want with their arm, and we would get back the corrected blood pressure absolute levels within the standards that are required medically by this organization called AAMI. And so that hasn't been done before in an unobtrusive way. And this is something that we're very excited about. It's a bit of a holy grail in the hypertension field. A lot of failures. We think we're starting a company that will work on this. And so more to come. We're hoping to offer something like this. And again, the challenge is people aren't actually measuring blood pressure even correctly. When you go to a hospital, you're only doing snapshots of blood pressure. A lot of times you're stressed out. And so you're actually measuring it wrong when you're actually, you don't have hypertension. It's also the other way around. There's something called masked hypertension, when you're actually not, with these snapshot measurements, you're not capturing it correctly. So by giving this to people's homes, we can actually assess, for example, how well the drugs are working, because we can do repeated measurements when they're kind of more in everyday environment, not sort of have, white coat syndrome or masked hypertension. So we're also, this is what Daiichi Shimbo, who is an expert in one of the world's experts in hypertension at the medical center. So both biochemical measurements and device-based for wearable devices. Let me move now towards the implantable work, also some of the therapeutic work, because the miniaturized devices are not just limited to sensing. They can also result in new therapeutics. And in fact, I think there's a whole paradigm out there where this is really underexplored. Miniaturized electronics has revolutionized really so many things in society. And I think the day is coming when it's going to also revolutionize therapeutics. You see some of this stuff with infusing, insulin pumps. You see it with bioelectronics, with neuromodulation, but with miniaturization, it's a whole different ballgame in terms of how we would even think about therapeutics, not just using drugs or biologics, really using devices. So I'm going to do this sort of in two parts here. One is using miniaturized devices, more with cell-based therapy. And then I will talk about the miniaturized therapeutics using bioelectronics. So if we start off with cell-based therapy, this is with Danielle Bajakian, who is a microsurgeon at Columbia. What we're working with is something pretty familiar to everyone at BME, organoids. But what we want to use organoids for here is actually for therapeutics. So we need a way not to make them in a chip and to do drug testing. We need to make them in massive quantities in a gentle way, and then we want to be able to inject them into the host for some reason. In this particular case, what we're trying to address is vascularization. We want to be able to form blood vessels, preform them in the organoids ex vivo, so that by the time we inject them into a region in your body that is ischemic, they're already preformed blood vessels. Because if you're injecting isolated cells, which there's all of these clinical trials on, for the severe ischemic cases, there's not enough time for these cells to be doing their work, and they're ischemic to begin with. So an ischemic region, so they basically die off before they can do their work. If we can actually inject them in a state where they're sort of have some blood vessels already preformed, we're asking them to just connect themselves, and we can treat these severe ischemic conditions. So the way this works is we have these microwells, and these microwells can be dissolved. And so that's sort of the basic idea. This is published again a couple of years ago here. I'll show some videos. These are microwells with endothelial cells and MSCs, and they contract. So it's just spontaneously, actually. There's no gel inside. The scaffold is an alginate scaffold, but the organoids themselves are completely cell-based. And based on, it's a self-organization of the endothelial cells and the MSCs. And then you actually get some sort of organization taking place where you can see some streaks here. So some blood vessel-like structures actually forming. And so you get these massive number of organoids that are being formed, and then now we're dissolving the alginate. And so it goes out of focus because now everything is dissolved. And then all of these organoids, self-organized organoids, can be injected into tissue. And you can see these blood vessel networks here that you can actually now into some tissue that they are forming. So that's the idea. Generate massive numbers of these organoids, in this case containing blood vessel-like structures, and then they can do their work once they are injected into the body. And we published this already. This is the method. I won't go into the details. And we also tested this in an animal model. This is a hind limb ischemia model in a mouse, and the left here, a hind limb of the mouse, is a control. The right one is the one where they've had a ligated femoral artery. So there's no blood flow there. But after 7 days, you can actually see the blood flow being restored. Typically, with just isolated cells, they typically require 3 to 4 weeks for you to see restoration of perfusion. And so here we're seeing it within 7 days. We are now testing this in more severe models of critical limb ischemia and trying to prove out this concept more. But I think the idea goes beyond ischemia. There's maybe a lot of other organoids that we can think about using this way therapeutically. Another possible application would be to treat obesity. This was not using this organoids approach, but it's another cell-based approach where we took, done with Judy Corner, also at Columbia Medicine, where we can take white adipose tissue and convert them to brown fat. The way you typically do this, I won't go into the whole excitement behind brown fat, but basically the idea is brown fat good, white fat bad. Okay, I'll put it that way. If you have more brown fat, then that could be a way to help with your general metabolism, but also reducing obesity and maybe diabetes as well. So you actually want brown fat. Brown fat is the good fat. And the way that people are thinking about increasing it endogenously is by drugs. A lot of these drugs have toxic side effects with heart side effects as well. So what we're thinking is, well, you know what? There's a lot of bad fat that people have. Let's take it, let's brown it ex vivo, which you have to do it in a bioreactor outside the body, and then we can re-inject that back into the patient, and you would have as much brown fat as you want. It's very localized, and there's not going to be this drug side effect. And so we worked on this, and this is published also 4 years ago now in scientific reports. So I won't go into that, but new ways of thinking about cell-based therapy using bioengineering approaches instead of drugs is, I think, pretty exciting, especially when you can combine the miniaturization element. Now, moving towards devices, this is something that was published now 5 years ago. We think you can even use hydrogels as a basis for forming devices. You don't need to use electronics. And so this whole device here is like using the mechanics of an expensive Swiss watch. It's called a Geneva design, completely mechanical design for how you spin the wheels of like these expensive watches. We built this all using hydrogels, which you normally think is squishy, hard to work with, but you can actually make it rigid enough to make this very intricate mechanical design. And here you can spin it wirelessly using a magnet. So this has been published, but we tested this and we worked on this with Francis Lee, who was at the time at Columbia. He's now at Yale, and he's a bone cancer expert. We were looking at osteosarcoma in real time. And depending on what was going on in the mouse, you can basically spin the gear and release locally a chemical chemotherapy. You don't have to release it systemically. So compared to the systemic lethal dose, we're actually 1/10 of that. And we actually get better containment of the growth of the bone cancer. So it's on demand, it's local release. Whenever you think you need it, you can actually then have this device, which is placed during sort of a normal process you would do with a biopsy, and release the drug, whenever you want. And you can do this completely with a 100% biocompatible hydrogel without having to worry about toxic, electronics. Now, you can also do this with ultrasound, which I'm going to talk about next. And this is focused ultrasound, which you can really up to a millimeter cubed focal volume, go deep tissue as well, and release drugs. We worked with Parag Chitnis, who is now at George Mason, on combining biomaterials with focused ultrasound so that we can tune the biomaterial so that it will be responsive to focused ultrasound, which leads us to the final topic of a closed loop device. And this is more for the bioelectronics, but also incorporating the sensing. So it's not an isolated drug delivery. Can we make this a hands-off device, almost like your pancreas for diabetes? It can sense glucose, automatically releases insulin. You don't have to think about it. And that's how our body works for homeostasis. We don't have to think about it. Our body senses things and we automatically adjust. So we don't have to think about diagnostics in one bucket, drugs in another bucket. Can we do all of this seamlessly using these latest technologies? And the project I want to talk to you about is done with Ken Shepard. This is a DARPA project, also with a number of BME faculty. There's a lot of faculty involved here, but in BME, also working with Clark Hung, with Helen Liu, and Milan Stojanovic, who's also with our department here. And so a lot of people here, but Ken Shepard is definitely the key person who's the bioelectronics expert, also in our department, and also a lot of other people outside of Columbia as well. The basic idea here is for wound healing. And the idea is not every wound is the same. So why are we treating it all in the same way by just applying some wound dressing to it? Yet every wound is different. And so if we're able to sense what is going on in the wound, let's say by looking at different protein concentrations in different parts of the wound in real time, we can also image it using ultrasound in our case, and just monitor what's going on in the wound. Can we feed that information into an algorithm that can interpret that information? And then that will say, this is the wound state you are in. This is what you should now do to accelerate the wound healing, either by releasing a drug in a certain part of the wound or by applying some other sort of stimulation. And it's an automatic, continuous process. And that way we will actually accelerate the wound healing. So there's 3 elements involved. There's the sensing element, there is the machine learning element, and then that will then teach how we will actuate to actually accelerate the wound healing. So it's a pretty ambitious closed loop kind of effort, and it's all coordinated by ultrasound. Now, how this works is the sensing part is using these very cool, very small motes that Ken has developed. He recently published this with Elisa Konofagu, who's another faculty in our department, expert in ultrasound. These are sub 500 micron CMOS chips. That is a full scale electronic structure, but they're super small. And they have no battery on board. They have a piezo element, so you actually can use ultrasound, focused ultrasound, to wirelessly power them on demand and actually communicate with them. Also on that chip is a field effect transistor, either one that detects pH, measures pH, or one that has an aptamer on it that can detect protein levels. And so whenever it wants, it's just sitting there, but whenever it wants, you would power it with focused ultrasound and say, hey, can you measure that protein level and then tell me where you are? This ultrasound patch that Ken is also developing is a wearable patch that is flexible, that also does not need to be moved. So if you're familiar with ultrasound, you usually have a transducer, you have to move it all around. This one here has an array of also piezo elements that can, through interference, focus in different parts of your body without the actual transducer moving at all. And so this thing just stays on you and it will steer the ultrasound to wherever it wants, to talking to whatever mode it wants. And then the transducer will then tell offline some algorithm, an algorithm will say, release some drug in a certain part of the wound. That's where my lab comes in terms of the work. This is just published in Angewandte Chemie by Rachel Field, who just graduated from our lab. And what we came up with here is a technique to make very reproducible hydrogel beads that are ultrasound sensitive. And what they are then is they can reversibly you can sort of just zap them with the focused ultrasound and they'll release a little bit of the drug. Now, the technique that people usually use for this is microbubbles. Microbubbles is something that is very short-lived. You have to inject the patient with microbubbles, they go in your blood, and then it's gone after a couple hours. And it's one time. Once you zap it, it bursts, it's gone. What we're talking about here is something that is more chronic. It can stay in this case, the wound. It can be reversible. Release a little bit. Now I don't want you to release it. 2 days later, I might want you to release some more. So the way we did this, first of all, with microfluidics, was to use these oil-water interfaces to, which I won't go into, to actually make these droplets. Now, there's a little bit more that goes into this that we published here. You can read about it. But it's a 2-phase hydrogel system. So there's actually 2 different phases, even in the water phase. And this is both PAG-based and a dextran. And so this is with the oil-water interface, you've got its droplet forming. But then within that droplet, where we have the 2 aqueous phases, there's 2 hydrogels in there. There's a PAG phase and there's a dextran phase. And so you can see they're very happy here. They're UV polymerized. as well. And I'll show you here, this is the setup for doing the UV polymerization and with the microfluidics. And then once you make these beads, they have this sort of core shell structure to them, which I'll show you. And then you can sort of zap them using focused ultrasound. They're responsive to focused ultrasound. I'll tell you why in a second. But what happens is then whatever compound is in the core phase of that hydrogel bead will be released from the core when you apply the focused ultrasound. So you can see the structures here. So you can produce a whole bunch of these beads. They have, there's a distribution, but it's a controlled distribution of both PEG and dextran. And that dextran core is when you will have that drug. And here, before the focused ultrasound and after, you can see actually there's a little bit of cracking and so forth in the structure. And we also see a telltale sign of this broadband noise in the acoustic signature, which tells us that there's a mechanism called inertial cavitation that's happening for those of you in ultrasound. And so we think that's what's happening when we apply the ultrasound to this. We're not seeding it actually with any nuclei. It's just sort of from whatever is around. But that's how the compound is released. And you can tell that there is a little bit of baseline release, which we can actually wash away. It really happens right at the beginning. But after that is mostly from the surface, actually, of the bead. But after that, you can repeatedly, stepwise, release these drugs over days using focused ultrasound. And so it's really, combining focused ultrasound with a biomaterial approach enabled by microfluidics in order to get this repeatable on-demand release. And now the thing about ultrasound, it can go deep tissue. So we think we can sprinkle this stuff in your body at some point. They're sort of inert, and then whenever you want, you can zap it, and they will release drugs on demand. Moving now to just a couple more things before I wrap up. We're also looking at how to help with the closing the loop part. And this study we also just published in Scientific Reports done by Priya, who just graduated. This is looking at interactions in a lymph node between T cells and dendritic cells. And there's a lot of reasons you might want to have a better method to identify an interaction between a T cell and a dendritic cell that involves a cognate interaction versus one that is just sort of a flyby non-cognate interaction when you want to screen for compounds that might be effective against as an immunotherapy and so forth. Or if you're into T cell engineering as well, you want to select the T cells that would actually work. Right now, the way for screening for this is based on biochemical measurements. And so it's a little bit laborious. You're also not necessarily picking up on all the T cells that might actually be recognizing your dendritic cell with a cognate interaction versus a non-specific one. And so we want to do this automatically. And this is using our favorite buzzword, machine learning. I won't go into this here. It's a relatively traditional neural network, so I'm not going to brag too much about that. But there are a couple of things that we found here. One is we actually added an antibody that actually helps with the discrimination between the cognate and the non-cognate. So it's not just a matter of doing machine learning. It's you can actually try to increase the discrimination by adding this antibody. We're not quite sure why it works. We have some hypotheses of adding this anti-CD40. And then the other part is by using the videos. And so it's not just with specific images, but by actually looking at the time series, we're also able to boost up the accuracy. Overall, our accuracy is over 0.9. This beats some of the other machine learning approaches, which hasn't been that many. but there was one published in Nature Immunology in 2019, and we beat them significantly here using this approach by both adding this antibody to further discriminate, and also by using a video series. And so, we're at 0.91, which is pretty good. The best so far, I would argue. Probably other people will do better in the future, but we wanted to come up, further contribute to this method of closing the loop. When you have a sensing screening mechanism automatically detecting what you want, and then and that's going to be the way to do it without user intervention. We also published this review of this whole idea of closing a loop, and maybe even incorporating robots that swim around in the body. By the way, I know that's a favorite topic of our chair at GLOW. And so we're working on that as well. How do you get things to swim around is one thing, but then you also have to have a way to control it and then to actually have it do something. And I think this whole idea is something we have been working on. Final topic before I close off and take questions here: entrepreneurial ecosystem, and so that's something I've worked on here. For those of you who haven't been up here at Columbia, for those of you who are here, so exciting what's going on in West Harlem right now. The governor of New York was here a few weeks ago, opening up a 350,000 square foot space, the Tasty building, around our multi-billion-dollar Manhattanville campus at Columbia. And so really, we're a lot of excitement. Obviously, great research right in this neighborhood, both the Morningside campus and Manhattanville campus, but now with real estate space opening up. Now, I had a bit of my part in this opening up an incubator called Harlem Biospace. We've been open that since 2013. On an academic level, just to talk about a lot of great things at the engineering school. I've been a co-director or director of the entrepreneurship program at the engineering school. These are all the things that are available to students and faculty in the engineering school related to entrepreneurship. It's a giant list. There's a website on this, and I think there's a lot that we can do. Now, if you want to step outside of the university, now right in this neighborhood, there's more and more of these companies. And I did, a little part here working with the New York City Mayors Office to open up this space for very early stage companies. There's now more and more companies. Just the other day, there's a company in this area here that just announced a $1 billion deal with BristolMyers Squibs. Now, our space hosts more early stage companies, the earlier stage companies. These are the companies that are currently in our space. We have 19 of them, and so not from all over New York City. So from the very small to some of these bigger companies, we're seeing that happen. And I also wrote an editorial here in Nature Biotech a number of years ago about biotech innovation. And so a lot of excitement academically and in industry. Now, also working with government partners, I actually left this off, but there's also a Pandemic Response Institute that I'm also working on with Shifu Chang and Andrew Smith, who's a smart cities person in engineering. We're working with this initiative that's led by Wafa al-Sadr, the public health, for New York City, the mayor's office, to try to prepare New York City better for the next pandemic response. And that's an example of not only academic innovation and even working with industry partners, but also working with the community. Those three elements, I think, are really unique in what we have in West Harlem. And it's really exciting for those of you who haven't visited here. I think we're doing something really special, even for the whole world. Finally, this something called Fourth Purpose at the university is something that our president, Lee Bollinger, really has led the way. This is something that is very unique, I think, among our peers. Now, a lot of the universities in the world try to take our knowledge at the university and make an impact in the world. It could be through translation, like startups, but it could be through other ways of making an impact in the real world through a variety of types of engagement. And but I think Columbia is really in the driver's seat here, thanks to our president recently I was appointed by our provost to be the vice provost for fourth purpose and strategic impact. Very proud to be doing that as a BME faculty member and as an engineer. Obviously, translation is going to be one of the key elements here. But I know a lot of us are in engineering because we want to make an impact in the real world. Columbia now is in a position where we're going to try to do this in a very cohesive effort, leveraging all of our assets intellectually across all our 17 schools at the university, as well as all the centers, as well as some physical assets as well. We have global centers all around the world, and both programmatically and with a variety of other means, we really want to not only publish great papers, which is great, but also work with partners, internal and external, to really make a positive impact on the world. So thank everyone in my lab as well for all their great work. Thank you very much.
Wow, thanks, Sam. That's really fantastic. Also, congratulations on being the vice provost for the Columbia University. I think that's a wonderful examples. We at Columbia will not just focus on each individual research. We actually care about the whole community, New York City, United States, as well as the global, the whole world as well. So we have some time for a couple of questions, and then I'm so glad to see our distinguished Professor Kam Liang is actively following your talk. So he said, great work. It is convenient to retrieval the implantable devices for drug delivery to the bone, which is supposed to, I know something about bone.
So the question is it easy to retrieve these?
Yeah, retrieve the implantable device from the bone.
Yeah, well, of course, Cam is going to ask the hard questions here. Now, on that particular, study, Francis Lee and I would argue that it's not a big deal to have that device there. It's biocompatible, and you're kind of going in enough times to do, I don't know, biopsies. Edward will correct me here, but whenever you're sort of in that environment, you can take it out as well if you want. But just to leave the device in there also is not the end of the world. That was also a fairly big device. With these smaller ones, then it's a matter of, if you're going the full hydrogel, whether it's going to be biodegradable or just biocompatible, maybe it's okay. Like we leave stuff in the body, on other devices, and it doesn't sound great, but it's not necessarily a problem. But yes, sometimes if you want to retrieve it, then we're actually working to try to retrieve these very miniaturized sensors and actuators, say using ultrasound guided retrieve or MRI guided as well. We are also exploring if you feel like you would need to because of toxicity reasons.
Great, thanks, Sam. So since we have been working on the COVID detections, and don't want to put it in the spot, right? So I just want maybe it's for public education as well. Is how I know when you do a home antigen test, right? In general, when you're positive, pretty sure positive, you're going to be positive by most likely from PCR test. But when you're negative, then does not mean you're going to be negative, right? You need to confirm by PCR. So what's the current data now in terms of one we received from the government, right? The home test kit is 50% accurate or 30% accurate?
Oh, it's not that low. If it was that low, you wouldn't be.
Okay.
No, it's probably at least 90% sensitive.
Okay.
You look at both sensitivity and specificity. When you look at this, I mean, what you're referring to is more sensitivity. It's probably 90% plus. And the CDC recommendation is to do, serial testing. So you test once, and then you can do it like a few days later. So, the issues is both in the technology itself, that it's not able to detect as high in the sensitivity, as well as just in the clinical manifestation. These protein markers, are different. And so there's some limitations there, but it's this trade-off between the performance and the accessibility that has always been discussed in the point of care space, that, I think we're all experiencing that. You can have the perfect test in the world, but if it's not accessible, it's okay to have an imperfect test if that's one that's going to be something that people can use, as long as you let people know how to use it properly. So just run it, as you said. If you're not sure, run it again, either right away or a couple of days after, is the CDC recommendation.
Thanks so much, Sam. And then we appreciate your Friday afternoons and give us the BME faculty in focus. And then stay tuned for our next speakers. Just please follow us, Columbia BME, on the social media. We'll give you update on what's going on. Thank you so much. Have a good weekend.